Okay, let's start. Hello, everyone. Good morning, good afternoon. So today we're going to cover new EN17141 norm, which is about biocontamination control. We will be conducting on microbiology in clean rooms and controlled environments. Before we start, if you have any questions, feel free to use this chat box and question session on your right or left side. Or if you are using an online version of GoToWebinar, still you can find this on the toolbox. So you can type your questions in anytime you want. So I'll be also monitoring this part. So if there's any questions that we can answer during the session, we will do. If not, I'm going to do it end of the session. If you don't have enough time, don't worry. I'm going to get back to you all of these questions right after this event. So let's start. My name is Haşim Solmaz, and I'm responsible for EMEA operations at Lighthouse Worldwide Solutions. And I have different heads in different organizations worldwide, but most of them are related to clean rooms and controlled environments. And I'm also participating member for the international organization, which is under ISO, Technical Committee 209, which were responsible for writing clean room standards. Today's webinar content will be about EN norm. I'm especially saying norm, not a standard, because this is a European norm, and 17141. The reason we conducted this webinar is because this EN norm is quite new and also will enlighten our future in terms of ISO standards. I'm going to come later to this part. But today we will be making an establishment and demonstration of environmental microbiological control parameters and the microbiological measurement methods and the guidance for life science, pharmaceutical and biopharmaceutical. Especially, I also added a medical device part. The reason I'm adding this medical device part is to show you how this norm is enhancing our quality in different applications. If you have this norm already, you will be notified that there is a special sessions for hospitals and the food and beverage industry as well. I'm not going to cover them today, but keep in mind, most of them are plus minus the same construction. So if you are in different industries, feel free to type in your questions. And one important part as a definition is a validation of air samplers. So in order to validate these air samplers, what we should do and what the manufacturers should do, and what are the difference between this validation other than having IQ and OQ protocols. Then of course, question and answer session. Okay. First part will be we'll be talking about the norm itself, the scope, relationship with ISO 14698, which is an international standard withdrawn from various countries, especially from the ISO perspective. And what are the relationship between this norm and the international ISO standard, and what's waiting for us in the future. First of all, this norm published in 2020, quite new, and prepared by the technical committee, SAN TC243, which is Clean Technology, the secretariat under British Standard Institute, Standardization Institute. And the document indicates a number of informative annexes. As you all know, if the norm or the standard is giving us information in an informative annex, this means that's not the norm itself. It's just giving a detailed information. If it's a normative annex, which means if you are following up this standard or a norm itself, then you have to obey these rules. So it is most likely a guidance in terms of application of microbiological contamination sampling. So the further guidance of biocontamination control in specific application. Reason I'm saying specific, it's not limited with pharma. As we said, this can also cover the medical devices, hospitals, food and beverage industry as well. So the scope of this norm here established the requirement and recommendation of methodology for microbiological contamination control. So we can say this established a plan, then in clause number 4, you can find this establishment in terms of microbiological contamination. Then implementation and operational activities also covered from clause number 4 to 5. Then monitor and review. Whatever you do, you have to double check and make a summary of this information, which is under clause number 5 and 6. To maintain and improve your activities, which is your reaction, is under 4 and 5 again. That's why the section 4 and 5, maintaining and implementation of your monitoring plan, microbiological monitoring plan, will be the main subject for today's webinar. This European standard supersedes the ISO EN14698, not the complete ISO 14698, the EN version of this ISO standard. And however, still EN17141, mostly based on ISO 14698, because expectation is not only covering the European regulations, also for the future, this will be the baseline for the future ISO regulations. That's why there is no conflicts with any regulation and standard. It was written carefully in order to not to introduce a new regulation, which will end up having conflicts with the current conditions. So the feature of EN17141 is to have a leading a new ISO standard, and this is already started. And this new ISO with a different coverage will also start it after these EN norm. And that's why we have new ISO technical committee 209 working group 2 formed last year during plenary meeting. So the same convener, Conor Murray, will be responsible for heading up this event and making a new standard available for all ISO member countries. If this is an EN norm, this means that this is only valid for the European countries. However, most of the international organizations are adopting this and implementing it, or it's still expecting, if you are importing your goods or exporting them to the European countries, then you should still be valid for these norms. So, establishment of microbiological control. We are going to start with planning steps, how we will collect samples from locations and what will be the frequency, our alarm and action limits, how we will react if we will not following these levels, and establishing a documentation system. Let's start with the baseline. First, there should be a microbiological contamination control plan, should be established, implemented, and maintained. This is a living structure, a life cycle, and it has to be verified after you have the final results out of your monitoring plan. And the system shall identify, control, and monitor factors that can affect microbiological contamination of the product. And of course, all the outputs that you are getting from the system, as well as the monitoring plan, has to be documented. That's the general summary of entire scope. Our steps for establishing a microbiological control is defined in this norm as: first, identify them. Second, it should be based on risk assessment. So make an assessment of your risk, take your notes, verify all locations, use a suitable risk methods like HACCP, FMEA, or fishbone analysis, and establishment of a monitoring schedule is important. Next one is establishing an alarm and action levels. This is not a one-time target. You have to verify after having these outputs versus your product sterility, and verification of continuing basis also covering this part. Establishment and maintenance of appropriate documentation. The final is education of a staff, training of a staff who are responsible to collect all these microbiological data, as well as verify them throughout the protocol. When we came into the risk assessment part, these are the important factors that we need to control. First of all, clean, controlled environment application. For example, pharmaceutical, medical device, food, cosmetics, or microbiological species. This can be a survival possibilities, associated toxins related to these species, and the potential for causing microbiological contamination of the product, or harm to the intended recipients. For example, if you are spoilage of product prior to the end of shelf life in food industry, for example, and product form, if it contains any preservatives or potential growth to substrates that may prevent growth, if you are using an intended product target population, this could be the infants or immunocompromised recipients, which are a high-risk patients. So you have to know all these details before conducting a risk assessment. Sources of contamination. Of course, the prime source are still people, the highly, especially the major source of contamination, not only microbiological, also for the particle levels, and the supply air system, HVAC system, recirculated or fresh air, whatever you are providing into the production line. Product materials itself, solid or liquid form, containers or packaging, and the utilities, compressed air, nitrogen, propane, oxygen, water for injection, they're all utilities that you have to verify as a source of contamination. Machines, of course, all the process and packaging equipment, the primary packaging infrastructure, sterile or non-sterile, still the machines are primary sources. Saturated sources, they're the secondary levels, but when you take a look at the first level, the prime sources, you're also covering all the saturated sources. For example, if you are covering the machine itself, of course, the surface of each machine or contact parts of this machine is also a controlled environment. Routes of transfer for microbiological contamination. This number one is airborne deposition. If there is a particles which can be subsidiary for the microbiological activities, these are all under airborne deposition. Surface contact, if the material or the human interface with the surface can be a high risk for microbiological contamination. All the liquid sources also listed under routes of transfer for microbiology. So when you conduct a risk assessment, you have to verify every single step here as a route of transfer. If your product or process has a specific routes of transfers other than listed here, it has to be included. These are all taken from the EN norm and listed here, but you can also consider other routes of transfer for your production specific conditions. So locations, it has to be under risk assessment based on a degree of risk. Reason we are saying here, as you can see, there are a list of locations that you need to control. For example, personal going and material transfers, product pass-through hatches. Consider materials and personnel that are moving. However, if you are adding more locations, you're also increasing more risk. More location doesn't mean less risk for your production, especially when it comes to microbiological control parameters. Reason is, first of all, when you are taking a sample from each location, you are interfering your infrastructure. Maybe there is no risk for the product itself, but you are introducing other risks due to your personal or your contact plate or moving into touching into these surfaces. So you are increasing your risk. That's why more location doesn't mean risk less. Less risk means proper planning, implementation, and control parameters in your infrastructure. So instead of increasing locations to be sampled, you have to focus on your risk parameters and degree of risk for each location. Again, the frequency of sampling will be controlled by the degree of risk, because adding up more frequent sampling, also making more touch into the surface, more interfere to the production line. That's why it has to be related to degree of risk. Specified in the microbiological environmental monitoring plan, it has to be written first, then will be implemented as part of your quality system. This could be a continuous monitoring or periodic, daily basis, weekly, monthly, or another agreed period. This has to be written again and will be implemented based on this information. Too frequent sampling could potentially introduce further risk, like adding up more locations. A good strike between balance between sufficient sampling and the potential introduction of contamination, you have to consider, because more sampling introduce more particles. Less sampling may require may increase your risk. So there should be a strike balance between these two conditions. Okay, alarm and action limits. First of all, alert levels will be lower than the action levels like we have on our microbiological particle monitoring systems, as well as here microbiological sampling. Appropriate target levels is important. If you are putting a low levels, it will take you into a lot of flags coming out of your system. So this end up with having more interfere with the production and introducing more microbiological agents into the production line. Or alert levels here to indicate that the microbiological contamination is higher than expected and give an early warning of potential loss of control. Every single alert level will give you a good indication of potential failure in the long run, maybe as an action level. So the specified alert conditions under which action to be taken, what you will be doing if you are getting these yellow alarms, which is our alert levels. When an action level is exceeded, immediate investigation is required to identify the cause. And if necessary, you have to take corrective actions. But taking it into consideration, one time exceeding this action level may not give any indication. That's why you have to use statistical calculations as well as trending, which will come later on. Average of your counts, standard deviation, distribution of your contamination, microbiological contamination, to get a better output from your infrastructure. Otherwise, one-time peak in microbiological sampling without investigation may cause a high risk, but having a huge investigation and making a lot of corrective actions to these one-time peak may end up having a lot of peaks at the same location due to a higher level of control. Documentation, of course, everything has to be documented, starting from the risk assessment all the way to get all this output and reviewed and updated as a living life cycle. And it's necessary to have and incorporate any changes implemented. So your documentation will really reflect whatever you are doing in production or in your laboratory conditions. Provide a review and analysis of microbiological environmental monitoring results. Any deviation has to be documented as well. Exceeded actions has to be reported, and actions taken to the corrected deviation should be also documented. So whatever you have in field has to be reflected into your documentation. Training is the most critical part, because having a good qualified and authorized people for collecting microbiological data, as well as making investigations, reviewing all the data from the field, requires a highly qualified microbiology trained people in field. They has to be competent for the microbiology. They should have a good understanding about what is microbiology, what is the growth, what is the reflection in terms of action or alert levels. So if you need further guidance, this EN norm addresses it into the ISO 14644-5, which is related to personal gowning and behavior. If you need additional guidance, for example, you can refer into these ISO standards. Demonstration of microbiological control. Let's start with this trending part. Microbiological environmental monitoring shall be analyzed, and the data trended in particular to identify an adverse trend. Trends should be analyzed at intervals that are frequent enough to identify any adverse trends. An adverse trend is considered more important than an individual result. Competent microbiologists should review the data to identify any possible issues. For example, an unexpected quantitative or qualitative shift in the types of microorganism has to be verified with these competent microbiologists. Trending is more critical than what you are collecting as a single basis. If you take a look at this graph, today maybe you are on the safe spot of your risk, but however, let me show you from the map here. However, when you have these trendings going high, this has to be investigated. The reason here, sooner or later, you might exceed the limits or something changed on your conditions. So you have to verify this information other than waiting to see the first red flag on top of your production. That's why trending and trend analysis with a good statistical calculation is the critical part of your production. Here, the verification. The result of microbiological environmental monitoring shall be examined periodically, and original limits may change after review of your current conditions. Maybe you can lower the limits in order to keep yourself in the safe side in the long run. It doesn't mean that if you're always within the limits, it doesn't mean that you don't have to do any corrective actions. Maybe you are seeing the trend is going high in a daily basis or in some particular locations. So you have to take precautions for the specific locations. And if verification indicates any deviation from the established limits or change, you have to take a corrective actions for these locations. Out of specs, these are important parameters to capture all these out of specs. But keep in mind, not all the out of spec conditions are the true results. That's why the competent microbiologists should verify that the out of specs are really contributing the data that you're collecting from field versus what you get as a result from the laboratory after incubation. That's why if there has been a real change on your conditions, it has to be determined and recorded into out of spec conditions. As a final part of your records, all the records has to be collected. All your instruments and internal audits has to be recorded. These records could be a documentation or a computerized system, but this has to be validated. And all calculations or statistical values has to be included. All the methods that you developed in order to get this data has to be in your records. Sometimes we are seeing that the end user coming up into the final result after sample tracking. However, their records are not really showing the data that they are collecting from field, or the way they are collecting data is not parallel to the records that they have. That's why it has to be up to date and reflecting what you are doing in field versus your records. Sample tracking. The sample analysis laboratory should have suitable and dependable procedures that allow for the clear identification and handling of samples from microbiological environmental monitoring. This shall cover the reception and progress throughout the entire analytical processes, and you have to get the final results throughout this sample tracking system. This can be an automated system and a fully automated system or a manual system, but it has to be documented again. And the correct identification with original monitoring location has to be defined. So the sample tracking system also being able to track the data from this location where you collect all the way to the laboratory result with one identification. Integrity of your results for demonstration of microbiological control. There are steps to avoid collection of false results. One is your application. Another is your identification of application-specific parameters, data collection locations, limit of detection of and sensitivity of test measurement systems, and the data collection points. Here for the data recording, all the data should come into the final location with the raw data, info held with this record, identification, as well as location of laboratory documents or a computerized records. If you are using workbooks, worksheet, or a computer, it has to be recorded within the same database. Procedures to be followed, for example, all recording, checking, correction, signage, counter signage of observations, calculation, and reports. Recommendation for consistent interpretation, and specific legal or regulatory requirement has to be recorded along with this data. Most of the data comes with just a colony forming unit numbers. However, if you are collecting data from field, it has to come with all these specific parameters. So if there is any deviation after statistical calculation or directly from one specific location, it should be analyzed with all these data recorded into the same path. Evaluation of your data. For each evaluation, a test procedure is required that describes the approach taken to develop the test method and the statistical techniques using to verify the test procedure. The agreed test procedure should be verified, and the risk shall be quantified and reduced to an acceptable level. And interpretation and evaluation of results should be based on more than one statistical method, and they have to be recorded. Reason we have interpretation and evaluation through the statistical value is because maybe you are getting one single high count in one which can be neglectable or due to error on your laboratory infrastructure. However, if you have a statistical method along with the trending, this will give you a right indication so that data evaluation should include at least one, but the best approach is having more than one statistical method. Trend analysis again. So again, single sample is not often more significant than having a trend analysis. Graphic representation is critical to identify the trends into our system. Control chart methods may be applied to provide an objective and statistically valid means so that you can verify all this information. Next one is microbiological measurement methods, which methods you are going to use to be sample and volumetric air samples and the culture media and incubation processes with an incubator. These are listed under microbiological measurement methods section of EN17141. First of all, choose of sampling methods. The sampling method selected shall be appropriate for the clean controlled environments to be monitored and shall take into consideration the following according to these EN norm. A, time and duration of the clean controlled environment activities. B, accessibility in the clean controlled environment for sampling device. C, effect of the sampling device on the process of or equipment to be monitored. D, efficiency and precision of the sampling method, which will be validated throughout the device. I'm going to conduct it later on. But here, I would like to add one thing into section B, accessibility of the sampling device. This is not limited with changing your Petri dishes for active sampling, for example, or putting these positive Petri dishes into the production line. You have to verify how you will take these Petri into the device before your production and how you will be changing them frequently without adding more risk into your production line. So accessibility should cover your device service requirements. For example, if you have this device inside your machine all the time, then you're going to introduce more risks. Or if your device doesn't have an HEPA output, then you are introducing additional risk into your clean room. So it has to be considered under this accessibility of your device. For volumetric air samplers, suitable sampling device shall be selected and supplier of the sampler shall demonstrate the collecting efficiency of the sampler, which we will be covering under validation section. The sampling techniques shall be validated. The volumetric airflow of the sampler shall be calibrated periodically so that you will be verified this information collected from device is true and you can trust on this device information. For the culture media, there are important points when you are selecting your culture media. The quality of this media shall be subject to suitable verification program and shall take into consideration the following points. Number one, justification of the selected media, for example, incubation conditions, detection of low levels of microbiological contamination. Second, establishment of a control For example, media will not contaminate it and not give you the false count. Third one, media suitability, based on the sterility of your media or ability to support the growth that you are targeting for your clean room or controlled environment. Use of neutralizers, especially disinfectant residues on the surface, does not supersede the growth of your microbiological activity on your sampling surface. Validation of the media used. You have to validate the duration of sampling. If you're taking, let's say, 36 minutes up to 4 hours, if there's a dehydration or due to airflow, or if this device or the sampling head impactor is not suitable for this, it has to be validated. Ability of the media to be detected, fungi as well as bacteria, has to be considered with this culture media. For incubators, the incubator shall be appropriately qualified before use and operation. Qualification here is what we generally call IQ and LQ protocols that has to be implemented before you start using your device. Thermal mapping is critical. If you are using a large incubator, you have to verify that the temperature is equal in every location and can be maintained throughout the incubation period. Requalification should be carried out periodically for this device, following service and other changes on your device. And temperature range in incubator shall be mapped plus minus 2.5% according to this norm with the targeted temperature. This qualification should be carried out at least once in every 3 years, like in our some of our clean room testing. Incubators shall be continuously monitored throughout the incubation period. Alarm has to be issued if there is any deviation from the required incubation temperatures. So this alarm can be integrated with a monitoring system so that if there is any issue with the temperature level or incubation period, then this alarm can be notified. Alarm can notify any of your personnel inside the lab or sending SMS, email, whatever required in order to take a necessary action. Next one is the guidance for life science, pharmaceutical, and biopharmaceutical applications. First of all, I can say this part of the norm is aligned with European GMP Annex 1. It's not giving a specific additional value to the application because they don't want to interfere with the GMP regulations. That's why whatever you know through the GMP Annex 1 still valid for this guidance. And again, for the limits of different zone and grades are given in European GMP Annex 1. This is the part of the section, also refers into European GMP. Reason here, as we said before, this device is not willing to get any additional value to the current conditions. And the reason is, if you are, let's say, giving a frequency or sample location number into the guidance, then the ISO standard or the GMP regulation will tell you different rather than this European norm. So adopting this norm will be not easy for everyone who are implementing currently an ISO 14698 as well as European GMP. But if we came into the medical device, the reason I took this medical device, it will give you a good highlight of establishing risk and how you will be collecting, where you will be collecting, and what will be the frequency of your microbiological control parameters. This medical device applications giving you the full scope of project. The reason I'm saying, in the long run, if you will have a new ISO, you should expect a similar approach with this ISO standard for microbiological control parameters, like we have this medical device approach. First of all, this EN ISO 14971 dealing with the risk assessment of medical devices, while 13485 is dealing with the quality management of medical device manufacturing. So these risk assessment steps are quite similar with the first part, where we also give the summary of this risk assessment. But there is one table comes with the annex of this part, giving you an example of microbiological risk assessment for a medical device product. Next one is to give a limit for a medical devices. If you take a look at table B1 on your EN17141 norm, this will give you a highlight here. Let me show you with a laser pointer. As you can see, this part, other than these categories, this part is completely the same structure with GMP Annex 1. So the category 1, 2, 3, 4 defined here is aligned with grade A, B, C, D, but just gives you the category for the medical device industry. Same here, number of locations to be sampled. This table B2 derived from the table where we used in ISO 14644-1, table A1, number of sample locations to be used for particle classification, cleanroom particle level classification. Only difference is if you are using active air sampler, you are going to take this N number and divide it to 3 with minimum of 1 according to this table. And if this is a surface sample, you have to add 3 and divide it to 3 to get these numbers from the formula table. If you are exceeding 1,000 meters square of area, there is additional formula given, like in ISO 14644-1. Again, this norm is not giving you a specific table, but making a new table out of 14644-1 table A1. Frequency of the sampling given in table B3, as you see here, if it's ISO class 5, 6, 7, and 8, by every shift for ISO class 5, for every working day for ISO class 6, weekly basis on ISO class 7, and monthly basis on ISO class 8. Keep in mind, this is not for pharmaceutical application, but this doesn't mean this is for medical device part. It's not for pharmaceutical production, but this doesn't mean that you can adopt it into your SOPs and give this table as a reference where you will be starting before you make your first risk assessment. You can use this as a starting point. And if you're not seeing any deviation for ISO class 5 by shift and ISO class 6 by working day, you can keep using them. And this could be a good baseline for starting a new production line without, if you don't have a history of your risk assessment, history of your data, which help you to make a risk assessment projection. Validation of air samples. There is two things quite important here: physical collection efficiency and biological collection efficiency. And we will take into consideration of air sampler revalidation process. First of all, validation of air sampler. Whoever sees this into the standard thinks that this is an IQ/OQ validation. In fact, it is not just an IQ and OQ validation. There are a couple of things has to be done by the manufacturer and a couple of things that has to be retained by the user. First of all, you have to determine the suitability of your air sampler depending on your application. And there is a physical efficiency, which is related to the ability of sampler to collect particles of various sizes, and biological efficiency, which assesses the ability of sampler to collect viable microbe carrying particles, which can form colony forming units. So this, due to complex and specialized activity required, the validation of sampler is likely to be carried out by the manufacturer, also through the third-party validation laboratories. So this validation is complex because you have to validate your sampling head, biocollector, which we call impactor, as well as your collection efficiency, physical and biological collection efficiency. When it comes into physical collection efficiency, this norm, EN norm, is referring into ISO 14698 cutoff rate, which is D50 value. This cutoff size, depending on the application, this cutoff size is the average equivalent diameter of microbe carrying particles that from the CFU are generally larger than 1 micron, and the D50 value smaller than 2 micron is considered appropriate. 50% means whatever you have on your air entering into this device has to end up at least 50% on your sampling plate, which will be healthy enough. You are not burning them into the sampling plate, and 50% is following the pathway, so that you are just taking at least 50% of the particles within between 1 micron up to 2 microns. For Lighthouse worldwide solutions, for example, our third-party laboratory results showing that the collection efficiency for 1.05 micron particles is exactly 50% collection efficiency. This is based on the third-party lab results, which gives us the D50 cutoff rate. This was valid for ISO 14698. So since EN17141 has also adopted this part from the previous ISO standard, this is still valid because the method is not changed. But when it comes to biological collection efficiency, this is the ability of sampler to collect viable microcarrying particles, and it includes the losses caused by both the physical collection efficiency and the effects that the sampling has on the viability of the microorganisms. This experimental work to determine the microbiological collection efficiency in 2 ways. One, aerosol chamber methods. This should be done by the third-party laboratories with living organism tests. Another is a simplified laboratory method, which is carried out by the end user as well, as a side-by-side testing your microbiological air sampler and give us a result, what you count from your unit under test and the reference unit. So both methods are defined under EN norm, under validation of air samplers. Because of the complex and specialized activity required, sampler validation is likely to be completed by the suitably competent external body, which will be under responsibility of manufacturer of active air samplers. The collection efficiency value and associated certificates should be retained by the user. So whatever you receive from your manufacturer, you have to verify this information and test it and adopt it into your system. So the retained has to be done by the end user. This is not the expectation that the validation work would be periodically repeated, because once it's done, either your impactor or the sampler system will be the same. That's why it's not required to be redone the same work unless you change the impactor structure or the air sampler structure. The sampler would be subjected to scheduled maintenance and calibration. This calibration could be done under the air velocity as well as flow testing, and the third-party laboratories as well as the production company itself can perform this. And the best practice here is to have ISO 17020 25 accreditation for making this calibration. So that's in general for validation of air samplers. Please do not mix it with IQOQ protocol. IQOQ is installation qualification and operational qualification, is where you are conducting in order to adopt this device into your production. But the validation of sampling is to verify that this device has a good biological and physical collection efficiencies. So these are 2 different individual activities that has to be done by the manufacturer. But if you want to do a biological efficiency side by side of your device, feel free to do it. Definition is made by the EN norm under this session. We came into the end. I would like to check if we have questions here. I can see a couple of questions. Okay, will you be able to share? Okay. Technically, in general, we have these technical questions. Okay, good. Check the chat box as well. Okay. One question is about the collection efficiency. Is this based on the Petri dish that we are using or the device itself? So the answer is device itself, because it is related to the single size of a hole on your impactor and the number of holes, as well as flow rate that you are using with your microbiological active air samples. On the validation of the test procedures regarding the laboratory. So verification and validation of the test procedures are defined in EN17141, and there are different tests has to be done. But if this is under the manufacturer's test, this has to be done with a third-party certified laboratories. Rise the action level. Okay. So if I have an area consistently falling, charging area, consistently falling, is it possible to rise the action level? It's not recommended. If you are not taking necessary precautions or necessary actions to make a root cause analysis, increasing your action level SOL is not a real solution. Your problem will be still there, but you'll be taking it under your level, action level. So technically you will be ignoring it. That's why it's critical to have a better understanding on how you set up your action level and how you will be reacting into these levels. Okay, is there a requirement for particle counter to be 1 foot from floor? For particle counter, I will answer this for particle counter and microbiological air sampler both. First of all, for particle counters, it has to be at the level of production or level of activity. If you're talking about level of activity, it has to be in a work level. So if your operators are walking inside the room, it should be around the shoulder level. We are generally taking 130, 140 from the surface, from the floor. But if this is for microbiology, if you're taking an active sampling, it has to be the same level based on the risk assessment. But if this is a contact plate that you are asking 1 foot from floor, still it has to be risk-based. For example, you can leave it to the floor if you want to see the deposition related microbiological activity on your passive Petri. But for active air sampling, we always use the production level or the room activity level as a reference point. What does the less than 1 CFU in the recommended limits mean? Okay, is it the average of counted colonies per plate or the actual counts per plate? Okay, very good question. The standards, especially the GMP, requires less than 1 colony forming unit in one location. The reason why we have less than 1, if you are considering one single location, it has to be 0 because less than 1 colony is not possible. But as you said, we're expecting it to be a scientific calculation based on statistical methods. So your statistical method can give you less than 1, larger than 0, as per your calculation. So if you are taking a lot of locations into consideration or one location throughout the trends, so your number would be 0 to 1 within between these two numbers. That's why less than 1 doesn't always mean 0. This could be a value of a statistical calculation. What will be the best culture method to use the air sampling yields and mold and general bacteria? Okay, this question is completely out of my knowledge because I'm not a microbiologist. The question was, what would be the best culture media to use for air sampling of yeast and molds and general bacteria? I'm so sorry, but I will check with our microbiology consultant, so to get back to you, Siraj, for this question. That's why I cannot answer it now. Good statistical tool to analyze trends and reverse alert and action limits. Would you suggest a good statistical tool to analyze? I think the best statistical tool is the standard deviation. Keep it simple. Reason, If your standard deviation is quite higher than your average number, which means you have a lot of peaks or you have a lot of out of scope ranges. But if you keep your limits into the certain level with a very small standard deviation, which means everything is under control and every single location has a plus minus same value output. And there are many other methods for making a statistical calculation, but keep in mind, all team members should have a full understanding on your methods. That's why if you are using, let's say, student T or other methods, statistical methods, you will make it more complicated. So keep it simple. Take the average, take the standard deviation, mean value, so that you will have a full understanding on what's going on in your clean room or controlled environments in terms of microbiological data. Okay, what is culture media? Okay. We have answered to your question, Mr. Haja. Mr. Alper Sarı answered your question, and he said that tryptok soy agar as the TSA is the best approach to use with your media, yeast and molds. Okay, good. Thank you, Alper Bey. Any other questions? I'm not able to see additional questions here. So we came to end. If you have other questions, you have my email here. I have my email on my presentation. Thank you very much for your time. I think we came to the end. If you have other questions, type them in. We can be in touch in LinkedIn, and you can also send your questions in. Thank you very much for your time. Hopefully, we will see you on our next webinar. Thank you. Have a great day. Bye-bye.